2006, a paper in the journal Nature documented something the field had been circling for years, but hadn't really reckoned with yet. Approximately 8% of the human genome, more than all protein coding genes combined, is derived from an ancient retrovirus. Not influenced by them and not distantly related to them, it's made of them. The viral sequences that integrated into the genome of our ancestors got copied into every cell of every subsequent generation and have been traveling inside us ever since. Most of it is inert sequences that integrated, lost function, and lost relevance over millions of years. Accumulated mutations, and a lot of them just sit in the genome as molecular sediment, kind of. Records of infections that had happened to primates, proto mammals, and earlier vertebrates across hundreds of millions of years. But not all of it's inert. Some of it is still running the placenta, the organ that made mammalian live birth possible, that made it possible to sustain a developing fetus for months inside of another body, continuously supplied, continuously supported, is substantially built from co opted viral sequence. The protein responsible for the most critical structural feature of the placenta is derived from a gene that retroviruses evolved to break into cells. That protein creates the fused cell layer that interfaces directly with the maternal blood supply and prevents the maternal immune system from destroying the fetus. The invasion mechanism became the protection mechanism. The infection became infrastructure. And as a consequence of that, carried forward across 130 million years of mammalian evolution is every placental mammal alive. And this episode is about how that happened, what the molecular record actually shows, and what it means for how we think about systems that encounter things that we're not necessarily built to recognize. To follow this, I'm Elizabeth, and this is Quirky Apes. To follow this story, you need a clear picture of what retroviruses are, and specifically what distinguishes them from every other class of virus. Most viruses operate on a hit and run sort of model. The virus enters the cell, commandeers that cell's replication, and it produces copies of itself and then either destroys the cell in the process or it exits to infect others. The virus's genetic material never integrates into the host's chromosomes. Your DNA, when the infection clears, is exactly what it was before. Retroviruses work differently at a fundamental level. They carry their genetic material as RNA rather than as DNA. When a retrovirus enters a cell, it carries with it an enzyme called reverse transcriptase, which converts the viral load of RNA. It carries with it an enzyme that converts the viral RNA into DNA. That DNA is then inserted into the host's cell's chromosomes by another viral enzyme. From that point forward, the viral sequence is part of the cell's genome. The cell replicates it. Every daughter cell inherits it. The virus is no longer behaving as a pathogen, it's behaving as a gene. HIV, for instance, is a retrovirus, and understanding how HIV works makes this a little more concrete often. When HIV enters a T cell and infects it, it integrates into that cell's DNA. It is doing exactly this, inserting its sequence into the host genome where the antiretroviral drugs can suppress its replication but can't totally remove it because it is now part of the cell's own genetic material. Every time that T cell divides, the daughter cell inherits that viral sequence. And that is why an HIV infection is currently incurable with our available treatments. You can't excise an integrated sequence from every copy of a genome in every cell of a body. The integration is permanent for the life of that cell lineage. Now, apply that logic to a reproductive cell and extend the scale from a human lifetime to hundreds of millions of years. In most retroviral infections, integration happens in somatic cells, the skin, lungs, liver, blood. Those cells eventually die, and the viral sequence dies with them. No transmission to offspring, no lasting evolutionary record. But occasionally, through the same infection process, a retrovirus integrates into a germline cell, sperm or an egg, or precursor cell that will become one. When that happens, the viral sequence is copied into every cell of every offspring and every cell of every offspring of those offspring indefinitely. The virus stops being a pathogen and starts being a heritable genetic element. These sequences are called retroviruses or ERVs. The human genome contains thousands of distinct ERV insertions, representatives of dozens of retroviral families that infected primates and pre primate lineages over the past 100 million years and more. End to end, they account for about 8% of the genome by length. Protein coding genes, the sequences most people picture when they think about DNA, those account for about 1.5 to 2%. We have been carrying more ancient viral DNA than conventional gene sequencing for the entirety of our species' existence. The sequencing technology to see it clearly didn't exist until the Human Genome Project. The analytical framework to understand what we were looking at took another decade after that to develop. The 2006 paper was part of that wave of work that forced the field to take the scale of ERV presence seriously, not as background noise, but as a significant fraction of the genome with its own functional history. Some of that functional history is still active. There is one of the most recently integrated retroviral lineages in human genome, with some insertions occurring even within the last few million years, has members that are still transcriptionally active in certain tissues and developmental stages. It's called the HERVK sequence, and it's expressed early in human embryos and in some cancers and in some placental tissue. The genome hasn't finished silencing all of it. Some of it is still running, and researchers are still working out what exactly it's doing. The early embryo expression is particularly interesting. In the first days after fertilization, before the embryo has implanted in the uterine wall, GRVK sequences are among the most active transcribed regions of the genome. The leading hypothesis is that the expression is not incidental. They play some regulatory role in early embryonic development, possibly relating to protecting that early embryo from other viral infections, or possibly related to establishing a new state of genome. The research is ongoing, but the pattern it describes is a genome that has been in active negotiation with a viral cargo for hundreds of millions of years, and that negotiation is not finished. The standard framing for ERVs, when they get mentioned in popular science writing, is often that they are leftovers, evolutionary baggage, junk with an interesting origin story. What the past 20 years of molecular biology have made clear is that framing was incomplete. ERVs are not just historical artifacts. They are a reservoir of sequencing that evolution has been drawing on across the entire history of placental mammals. And the most important thing it has drawn from that reservoir is the placenta itself. Before placentas existed, vertebrate reproduction was constrained by what an egg could hold. An egg is a closed developmental system. Every calorie, every protein, every signaling molecule the embryo will need between fertilization and hatching has to be loaded in before the shell closes and the membrane seals. So the resource budget is fixed at the start. There is no mechanism for mid development supplementation. That is a workable constraint. Fish, amphibians, reptiles, and birds have all evolved remarkable solutions within it. Elaborate yolk compositions and carefully timed hatching with extended parental provisioning after the fact. But there's still a ceiling on the neurological complexity that a fixed resource egg just can't exceed. The human brain, on the other hand, requires roughly 25 years of active construction and uses about 20% of the body's resting metabolic energy in adulthood. Building that brain, the gestational phase of its development requires continuous high bandwidth metabolic support for 9 months that can't get preloaded into an egg. The placenta solved this by making that embryo metabolically dependent on the mother in real time. It is an active organ, not a passive scaffolding, and it maintains continuous bidirectional exchange between the developing fetus and the maternal circulatory system. So, oxygen, glucose, amino acids, hormones, immune factors, they all flow toward the fetus. Carbon dioxide, metabolic waste flows back. For the full duration of the gestation, the fetus is running on the mother's metabolism. The placenta is the interface that makes that possible. And without it, the brain that's sitting in your skull could not have been built. The immunological problem this creates is severe and it's often underappreciated. A fetus isn't genetically identical to its mother. Half of its genome is paternal from a genetically distinct individual. The maternal immune system is specifically designed to detect and destroy genetically foreign material. That logic drives organ transplant rejection, and it applies here directly. The fetus is immunologically a foreign object, and it should read as a target. It should be rejected. That is that it mostly is not rejected is one of the more remarkable facts of mammalian biology, and the full explanation is still being worked out. Multiple mechanisms are involved, including local immunosuppression at the maternal fetal interface, modified expression of cell surface proteins on fetal tissue, and active immune tolerance mechanisms. But a central structural piece of the explanation is the architecture of the placenta's outermost layer. This outer layer is unlike any normal tissue in the body. It's not composed of individual cells. It's what they call a syncytium, a continuous multinucleated sheet formed by a complete fusion of cells into a single shared membrane with no cell boundaries remaining. This structure presents a surface to the maternal immune system that doesn't trigger standard rejection in the way a tissue composed of discrete fetal cells would. This is also the physical interface through which that placenta contacts the maternal blood supply. Its structural continuity isn't incidental to the function, it is the function. Creating it requires fusing cells together at a scale and maintaining that fusion throughout gestation. That is a specific and a very non trivial biochemical problem. The protein that solves that is syncytin. It's derived from a gene of ancient androgynous retrovirus. The ENV gene encodes the envelope protein, the protein on the viral surface that allows the retrovirus to fuse its lipid membrane with the host's cell's membrane during that infection. The membrane fusion is the mechanism by which the virus gets inside the cell. It is specifically an invasion tool. And somewhere around 130 million years ago, a mammalian ancestor with an integrated retroviral sequence containing a functional ENV derived gene had that sequence expressed in blast cells, the cells that form the placenta. Those cells fused. And More cells were formed. The maternal immune system didn't destroy the embryo. The lineage survived. There is an additional layer worth naming. Syncytin does not only drive cell fusion, the viral envelope protein also carries immunosuppressive domain, a region that actively dampens local immune activity at the vicinity. Retroviruses evolved this immunosuppressive capacity for themselves. It means they can suppress the host's local immune response long enough to replicate. In the placenta, that same capacity protects the fetus from the maternal immune system. The virus's immune evasion mechanism is now the fetus's immune shield. PEG10 is a gene derived from a retroviral gag sequence. It's essential for the earliest stages of placental development before those blasts even form. Mouse embryos with PEG10 knocked out failed to develop a functional placenta and they die at mid gestation. Multiple ERV derived regulatory sequences also control the expression timing of placental hormones, including those that regulate the onset of labor. The placenta is not tangentially connected to the ancient viral DNA. It's built from it at multiple levels in its architecture. And those contributions aren't redundant. If you remove them, the structure itself fails. The downstream consequences of that structure play out in the fossil record. When the Cretaceous Paleogene extinction event eliminated the non avian dinosaurs around the 66 million years ago mark, the ecological niches that they had occupied were suddenly open. Placental mammals, small, relatively obscure mammals for the Mesozoic, diversified explosively into those niches over the following 10 to 15 million years. Every major mammalian lineage today traces its origin to that radiation. The placenta is what made the nervous system complexity of that radiation possible. The placenta is built substantially from ancient viral DNA, and the mammalian world, as it currently exists, in a direct and traceable sense, is a consequence of retroviral infections that happened to vertebrate ancestors during the Mesozoic. That is the exact sequence of events. It's not a metaphor, and that's not an overstatement. Mammals didn't just discover the solution once. There's a lizard that gestates live young with a nutritive placenta, extended gestation, active maternal fetal nutrient transfer, and a specialized interface layer of analogous that's analogous to the mammalians. And this is not a reptile that retains eggs inside of its body until hatching, which is a separate adaptation that multiple reptile lineages have evolved. This specific animal has a genuine nutritive placenta derived from egg laying ancestors evolved completely independently of the mammalian lineage. When research ex- when we when researchers examined the molecular basis of that placenta, ERV derived sequences were involved in its function. Different retroviruses, different evolutionary trajectory, but the same structural solution. Preliminary evidence from certain live bearing fish point in the same direction. What this convergence indicates is that the mammalian case is not a singular, improbable event. Retroviruses have been infecting vertebrates for hundreds of millions of years, depositing different envelope proteins into vertebrate genomes across the entire span. When vertebrate lineages independently arrived at live birth with nutritive placentation, they had access to the same raw material, those proteins that were already present in the genome, and they kept finding it useful for the exact same purpose. The same molecular tool, independently co opted, producing analogous structures in unrelated animals. There's a researcher at Oxford that described the logic directly. Viral proteins already have functions. It is much easier to borrow these than evolve them from scratch. The retrovirus had solved a specific engineering problem: reliable membrane fusion under set physiological conditions, under their own selection pressure, and for their own purposes. But that solution, deposited across vertebrate genomes through millions of germline infections over a deep evolutionary time, was available to be found. Multiple lineages have found it independently. The placenta is what happens when you find it, and the selective conditions for live birth are in place. This is convergent evolution operating not on gross anatomy or behavior, but on co opted genetic sequence. It is a different register of convergence than the classic examples, like the independent evolution of eyes or wings or streamlined body plans. It is convergence at the level of molecular raw data, and it suggests that what happened in mammals was not lucky. It was, given sufficient time and retroviral diversity in the genome, very predictable. The genome did not receive those retroviral integrations as resources. The cell defense mechanism that went into work on them went in immediately. There's a DNA histone modification and a small RNA based silencing pathway that all function to identify and suppress foreign sequences. The default response to an ERV insertion is containment. That containment mostly held. The majority of ERV insertions in the human genome are silenced. Heavy methylated, transcriptionally active, gradually accumulating mutations that erode their sequence toward eligibility over millions of years. They are under control. The genome suppression mechanisms are doing exactly what they are designed to do. What happened with the Cynstin lineage and with the PEG10 and with other ERV derived sequences now perform essential placental functions. It's more specific than general failure of suppression. The silencing was tissue specific. These sequences retained enough functional integrity to be expressed in trophoblast cells, while remaining suppressed elsewhere in the body. How that tissue specific escape from silencing happened, whether it was through gradual erosion of silencing signals or through positive selection for expression there, or through some combination. That's still an active research question. But what is clear is the viral sequence that the genome had been managing as a threat became expressed in exactly the tissue where its function turned out to be structurally necessary. It is important to be precise about what this process was and what it wasn't. The genome does not decide to use the viral sequence. There's no technology here. What happened is over millions of generations, organisms that happen to be expressed in the trophoblast tissue and in which that expression produced better placental function left more surviving offspring than mechanisms that didn't. The viral origin of the tool was irrelevant to that selection. The selection pressure was entirely on the functional outcome. Did the placenta work well enough to sustain fetal development through gestation? The majority of ERV sequences never found that window. They were suppressed completely, they mutated beyond functional relevance, and they became the molecular fossils that make up most of that 8%. For those sequences, suppression was the correct outcome. What makes the Simpson and the PEG10 cases remarkable is not that the genome was latent, it is that the specific tissue context, under specific selection pressure, sequences that had been treated as foreign turned out to do something different, turned out to be doing something the genome had no other efficient way to do. The innovation came from the gap between the suppression mechanism and the conditions under which that suppression was incomplete. The gap over deep evolutionary time is where the placenta was built. The reason this story belongs on this show is the biological processes and the social processes I work with are not the same thing. And treating them as a tidy parallel would lose the actual argument. What biology provides is the language for a pattern that is otherwise hard to name without sounding like advocacy. Systems maintain coherence by managing anomaly. And that is not a flaw. It is exactly how these systems stay functional. A genome that could not suppress foreign sequence insertions would be destabilized. An immune system that couldn't distinguish itself from its non self would destroy the organism. An institution that could not route around an individual variation would lose the coherence required to function. Anomaly management is load bearing architecture in biological and organizational systems alike. The problem is not a management function. The problem is what happens when that function has no mechanism for distinguishing between two structurally different situations. One in which the anomaly genuinely has no useful function in the current context, and one in a system has not yet encountered the right conditions to discover. When those two cases are handled identically to suppress, contain, or route around, the system is trading adaptive potential for short term coherence. The cost is invisible because it never gets tested. In my work with neurodivergent people and the organizations they operate inside, this plays out in consistent structural patterns. The traits that organize non normative cognition get treated as different, as deviations from the functional baseline. The accommodation framework built for neurodivergent employees are structurally containment frameworks. Here is how we will manage that deviation's impact on the system. The system's central question is how to minimize disruption. That is a different question from what does this trait actually do? And what would it take to build something that uses it rather than works around it? A concrete version of this is when I work with organizations on team design and workflow architecture. A recurring scenario involves a team member who is highly effective at a specific class of problem, say identifying failure modes in complex systems, or maybe generating non obvious solutions under ambiguous constraints, and or even holding a large amount of structural complexity in working memory simultaneously. These are genuine capabilities. The team depends on them and can't easily replicate them. The same person struggles significantly with meeting cadence and status update requirements and linear documentation processes in the team's workflow that's built around. The standard organizational response is performance management process that focuses on compliance gaps. The gaps are real and the processes address them. The capability, which is also real, and the team has no other way to reproduce, doesn't enter the analysis. The system pays the cost it hasn't even named in a currency it's not even tracking. The intervention I usually bring is not to eliminate performance expectations, but it's to run the functional assessment that the system skipped, to ask, what does this trait actually do? Under what conditions does it do it? And is there a structural design solution that creates those conditions rather than simply documenting the failure to meet requirements that were built for a totally different kind of nervous system? Sometimes the answer is that the mismatch is real and the role is wrong. But often the answer is that the system has been applying the suppression response to something it's never actually examined, and the examination changes the options available. The same pattern runs through educational contexts. A student who produces inconsistent written output, but is exceptional in some other assignments, absent or disorganized in a few, gets routed into academic support processes that focus on inconsistency as the primary problem. The implicit model is that consistent performance is the baseline and inconsistent performance is the deviation to be corrected. What that framing does not ask is what the conditions were for the exceptional output versus the absent output, and whether those conditions are reproducible by design. When you ask that question, you often find the inconsistency isn't actually random. It is a legible signal about the conditions under which that particular nervous system can and cannot produce. That is actionable information. A system organized around suppressing the signal rather than reading it isn't running a more rigorous process. It's running a less informed one. Neuroinclusion, as I practice and teach it, is about building the assessment mechanism into the system architecture. It's not a compassion initiative, but it's a functional one. The ERV record does not prove that neurodivergent traits are universally valuable. That's not the claim. The claim is structural, that systems that cannot tolerate unrecognized input long enough to assess it are paying the cost in foreclosed potential that they've never even accounted for. Most of those cases, like most ERV insertions, probably won't produce something essential, but some of them will. And the only way to find those will be to build systems with enough structural patience to actually let assessments run. And the late identified experience has a specific texture that is hard to describe accurately without either over dramatizing it or flattening it into something pretty generic. You've spent years, often decades, building workarounds for a nervous system that you probably didn't have accurate information about. And some of those workarounds were probably genuinely good, adaptive and efficient and durable, but they were all organized around the same implicit goal, making the deviation less visible. Perform the baseline well enough that the system does not route you into the management process. What that does over time is produce a very detailed, very well maintained map of the ways your cognition does not match the environment you're operating in, and almost no map at all on what your cognition actually does when it's not spending its resources on that performance. Getting an accurate diagnosis late and then doing the actual work of understanding what that diagnosis means neurologically rather than just categorically involves rebuilding that map. Instead of I have attention problems, you might learn that attention is not a deficit, it's differently allocated, intense and largely involuntary rather than modulated and voluntary, directed by salience and interest rather than priority hierarchies and scheduled task lists. That is a different functional profile than clinical language often suggests. Rather than I'm oversensitive, try sensory processing operates at a lower threshold and higher resolution, which often produces both an overload problem and the pattern recognition capacity that underlies a significant portion of what I do effectively as a practitioner and a consultant. The retrovirus model gave me a frame for something that I'd been trying to articulate for years without quite getting there. That protein in Synuclein did not become a different kind of protein when it was recruited into placental function. It kept doing exactly what it has always done, fusing membranes. The function didn't change. What changed was whether the surrounding architecture was built to use that function or suppress it. The traits that characterize my neurology are not degradations of a more correct underlying system. They are features of how this particular nervous system is organized, with costs, with functional capacities, and with conditions under which each shows up or does not show up. How do we manage this person's neurology? And how do we build an environment in which this neurology functions are not the same question. They're not even about the same problem. They produce very different designs, very different outcomes, and a very different relationship between the person and the systems they're working inside. And that distinction is the practical center of my work, not as a value position, but as an engineering one. One of the concrete effects of late identification is that you often spend a significant portion of your adult life working inside systems that were running the suppression response on your traits before either you or they had any accurate information about what those traits really were. That may not necessarily be a grievance as much as a data point. Most of the people I work with have the same data point, and most organizations they work inside have no idea they are paying for it. The cost is distributed and invisible in the way that foregone potential always tends to be. You don't see what didn't happen, you only see the deviation that got managed. This work, in part, is about making the cost visible enough to be addressed. The sequence of events, roughly 130 to 160 million years ago, a retrovirus integrated into a germline of mammalian ancestor. That sequence was inherited over millions of generations. The genome silencing mechanism failed to fully suppress a specific viral ENV gene in trophoblast tissue. The gene encoded a protein, and that protein fused trophoblast cells into other blast cells. And the resulting structure allowed the fetus to access the maternal circulatory system without triggering immune rejection. Extended gestation became possible. Complex mammalian neurology became possible. The Cretaceous Paleogene extinction event wiped out the non avian dinosaurs around 66 million years ago. And the placental mammals, already carrying that viral infrastructure, diversified to fill every ecological niche available. The lineage eventually produced, among other things, the species having this conversation. You exist because a foreign sequence was not completely eliminated. Suppression responses aren't wrong. The point is that suppression is not assessment. It is fast, it operates on pattern matching, and it runs before the functional analysis has occurred. In biological systems, the gap between suppression and assessment is bridged in evolutionary time, deep time, during which the minority of cases where suppression was the wrong call eventually surface through selection pressure. Organizational systems don't have that length of time, and they don't have an automatic selection mechanism that surfaces those cases. They have to build one deliberately.